everyone. So we are here for another class. In this we'll be discussing on carbohydrates in elective 1, unit 1. Now first let's see what are these carbohydrates. By definition, carbohydrates are polyhydroxyaldehydes or ketones or substances that yield such compounds on hydrolysis. Now what is the meaning of that? You know, poly means many, hydroxy means OH group. So polyhydroxy means many OH groups are present. That is one thing about carbohydrates. The second one is either they are aldehydes or ketones. If the functional group is CHO, it is aldehyde. If it is C double bond O, it is ketone, isn't it? So carbohydrates are polyhydroxyaldehydes or polyhydroxy ketones or any such substance that yields these kind of polyhydroxyaldoses or ketoses on hydrolysis are called as or can be called as carbohydrates. Most of these carbohydrates have empirical formula of CH2O n times. For example, glucose C6H12O6. Okay, so where C6H12O6, if you write the empirical formula, it will be CH2O 6 times. N is equal to 6 there. Okay. It may also include the other derivatives that are produced as a result of hydrolysis. Now, these are most abundant organic molecules, okay, and they are also called as saccharides. And the word saccharide is derived from Greek saccharon, meaning sugar. So carbohydrates that are simple, that can be that are soluble in water and sweet in taste are called as sugars. Okay, now each carbohydrate molecule, it's formed of carbohydrate. So as the name itself says, it is a hydrate of carbon. Okay, that means it should contain hydrogen and oxygen, hydrate, water. So carbon. So that means carbohydrates are basically formed of carbon, hydrogen and oxygen. Okay, so what you see there on the screen, we have two examples there. One is glyceraldehyde and other one is dihydroxyacetone. So in both the cases, see, by definition it is a polyhydroxy compound. You can see at least 2 OH groups there, isn't it? And then polyhydroxy either aldose. See the first one, glyceraldehyde, C double bond OH, aldehyde group. Second one, C double bond O, it is a keto groups. So by definition, polyhydroxy aldoses or ketoses are called as carbohydrates, and basically they are formed of carbon, hydrogen and oxygen. As you can see there in the picture, there is carbon, there are hydrogen atoms and there is also oxygen. And now all these carbohydrates are basically formed of simple sugars called as monosaccharides. Moving to the classification of carbohydrates, carbohydrates are basically classified into different types based on number of sugar units present in them. So the first class is monosaccharide. Saccharide, as we all know, means sugar. Mono means single. So monosaccharide means single sugar, or they are formed of single sugar molecules which cannot be further hydrolyzed. Okay. Next one is disaccharide. Here oligosaccharides and disaccharides can be clubbed together under one name as a oligosaccharide itself. So I'll go with that classification, where oligo means few, saccharous again means sugar. So here any carbohydrate that is formed of 2 to 10 sugar molecules, okay, then you call it as oligosaccharide. And if it is formed of 2 sugar units, okay, then it is called as disaccharide, 3 sugar unit trisaccharide, 4 sugar unit tetrasaccharide and so on. The last class of carbohydrate is polysaccharide. So when more than 10 sugar units are present, then it is a poly. Poly means many, saccharide means sugar. So many sugar units form polysaccharides. You can see the examples also. We will be seeing them one by one in the later slides. Now let's see monosaccharides. As we all know, mono means single, saccharides means sugar. Monosaccharides means those which are formed of single sugar units, and generally they consist of single polyhydroxyaldehyde or single polyhydroxy ketone unit. Okay, and these are generally called as simple sugars because they cannot be further hydrolyzed. Now, what is this hydrolysis? We are frequently using the term. Hydrolysis means it is by breaking a molecule of water, okay, or by using a molecule of water. If a bond is broken by adding hydrogen and OH across the bond, then it is called as hydrolysis. Hydro, water, lysis, breakdown. Breakdown of water results in breakage of the bond. That reaction is called hydrolysis or hydrolysis. So, by such a reaction, if it can be broken down into further units, further simple units, then it is not a monosaccharide. If it cannot be broken down further by hydrolysis, then it is a monosaccharide or simple sugar. Okay, these monosaccharides or simple sugars are generally colorless and they can be crystallized, okay, and these are generally soluble in water and are insoluble in non-polar solvents. Now, these monosaccharides, their backbones are formed of unbranched carbon chains, okay, and these might vary from 3 to 7 carbon atom. Don't get confused. In previous slide, we saw the classification where oligosaccharides we have seen. Oligo means few, saccharide means sugar units. The number of sugar units will give the name disaccharide, trisaccharide, tetrasaccharide. There we are talking about sugar unit. Here we are talking about number of carbon atoms. We said monosaccharide is a simple unit, so it cannot be further broken down, but it is formed of number of carbon atoms, just like glucose, C6H12O6. So here number of carbon atoms are 6. Okay, so based on the number of carbon atoms present in the monosaccharides as well as based on the functional group present, these monosaccharides are further classified. And here, while naming, the common suffix used is ose, glucose, fructose. Okay, so ose is used as a suffix. Now, if a carbonyl group is present at the end of the carbon chain, you can see the first diagram again, glyceraldehyde, which is a aldotriose. If it is C double bond O, it's a carbonyl group. Okay, if it is present at the end of the carbon chain, then it is an aldehyde group. You are familiar with the aldehyde group, C double bond OH. CHO is an aldehyde group. Then it is called as such a monosaccharide that contains a aldehyde group is called as aldose. And if the carbonyl group is present within the chain, see as you can see in the second one, dihydroxyacetone or in ketotriose, here the carbonyl group is present within the chain and that's why it is a ketone group and the monosaccharide is called as ketose. So here glyceraldehyde is a aldose and dihydroxyacetone is a ketose. Okay, these are the simplest monosaccharides. Now going back to the classification of the monosaccharides, I said monosaccharides are further classified based on the number of carbon atoms present in them. Okay, so if there are 3 carbon atoms, triose, example glyceraldehyde or dihydroxyacetone. Okay, if there are 4 carbon atoms, tetrose, tetra means 4, OSE, it is a suffix for identifying a monosaccharide. Example is erythrose or erythrulose for a ketose. Next 5, penta means 5, so pentose for ribose, okay, and hexose, glucose is an example, and even you can take the example of fructose or galactose. All these are formed of 6 carbon units. So I will repeat, classification of monosaccharide, they can be further classified into triose, tetrose, pentose, hexose, heptose, etc. based on the number of carbon atoms present. Here I have concluded only up to 6 because these are the major type of monosaccharides that are generally present in the living system. There are few more anyway. So common monosaccharides that are present in living system are glucose, fructose, galactose, ribulose or ribose, deoxyribose, that is a derivative of ribose. Okay, and here we have the pictures of some aldoses and as well as ketoses, D-glucose, D-mannose, D-galactose. These are the isomeric forms of each other. You can see the difference. only position of OH group is different in 3 of them. Then there is a fructose. Fructose is also a isomer of glucose, but it is a functional isomer. And we have ribose, that is a pentose. You see there are now only 5 carbon atoms are present in the backbone. Okay, so these are few monosaccharides. Again we have picture of hexoses, glucose, fructose and galactose. If you see, fructose looks like a 5-membered ring. No, it is actually 6-membered one, but CH2OH is present outside the ring. Okay. So glucose and galactose are again isomers, and here the difference is that the position of OH group, okay, in the first carbon atom. In case of galactose is present above the ring, in case of glucose it is present below the ring. Okay, first carbon atom, that's a major difference what you can find there. Okay, next one, pentoses. Here we have the example of pentoses, 5 carbon containing compound it is. Okay, so in the ring there are 4 carbon atom, and above the ring there is one more, 5th carbon atom is present. And the difference between the ribose and deoxyribose is that you observe the second carbon atom there. The second carbon atom has OH group in ribose, and in deoxyribose it has only H. Deoxy, one oxygen has been removed, that is the meaning of it. So deoxyribose sugar is a pentose sugar where one oxygen atom has been removed in the second position of the carbon, okay, from the second carbon atom. So that's what deoxyribose is. Now let's see the different biological significances of all these different monosaccharides we have discussed so far. First one is glucose. You can see the skeletal structure of glucose, C6H12O6, where the OH groups are present above and below the ring. Okay, there are different ways of writing the cyclic structure of these carbohydrates, sugars. What you are seeing here is a pyranose ring form. Okay, so glucose is a monosaccharide formed of a single sugar unit, and it is a naturally occurring sugar, and it can form combine with either itself, that means glucose, number of glucose molecules can combine with each other, or it can also combine with other monosaccharides to form disaccharides to polysaccharides. Okay. And glucose, in case of humans, not only in humans, in most of the organisms, it's very important biologically because it is the important nutrient for fueling the body. Okay, and it is also important because our brain and nervous system get the energy in the form of glucose, and this glucose is broken down in glycolysis, Krebs cycle, and ATPs are generated. You all know that ATPs are the energy currencies of the cell. And this is also, as I said in my previous slide, they form large number of oligosaccharides and polysaccharides. So when two glucose molecules combine, they form maltose. When one glucose and galactose molecule combine, they form lactose. And when glucose combines with the fructose, it forms sucrose. Like this number of oligosaccharides as well as polysaccharides, like cellulose. Cellulose is a polysaccharide formed of, it's a polymer of glucose. Thousands of glucose molecules form a cellulose. Similarly, starch and glycogen, they are also are the polymers of glucose. Not only these, there are hundreds of different derivatives of glucose. In one or other form, it forms a large number of structural and functional components of cell, tissues and organisms. So, glucose is one of the very important monosaccharide which is a primary fuel providing energy to the cell as well as to the body. Okay. Next monosaccharide is fructose. So, this fructose, it is the sweetest carbohydrate that is available, and the plants makes lots of fructose so that they can attract the insects. So, these are generally present in fruits. So, they are also known as the fruit sugar. Okay, it's also known as a fruit sugar. So fructose is a commonly occurring monosaccharide in case of fruit. So even the honey contains fructose. Okay, and the major biological function of the fructose is that it can act as an alternative metabolite in generating the energy. Okay, it can be alternative for glucose in the sense it can actually enter into the glycolysis. In detail we will be studying glycolysis in one of the coming chapters, and you might have already studied them in your previous classes also, where glucose is broken down into pyruvic acid. So in the same pathway, fructose can enter and can produce intermediates that might helpful in cellular respiration. So it can also enter into other metabolic pathways like glycogen synthesis. Glycogen is a polymer of glucose. So in the body, glycogen synthesis can occur by the fructose. So triglyceride synthesis and free fatty acid synthesis, that is the synthesis of lipid, and gluconeogenesis. So this is a process where glucose is produced in other than the normal methods. So when there is a necessity of glucose and normal glucose is not available, body switches on to other mechanisms by which acetyl coenzyme A will be generated, either by using some amino acids or the fat that is present, okay, or even by using the fructose. Gluconeogenesis can be done. Okay, so that body gets what is the necessity of glucose, ultimately getting the energy in the form of ATP that can be generated by a process called as gluconeogenesis. Okay, so fructose is useful in such reactions also. Okay, and it can also be used during glycation, where a lipid or a protein is combined with the carbohydrates. So in protein and lipid you will come across complex proteins and complex compound lipids. There will be studying about the carbohydrates that combine with the protein, okay, glycoproteins or glycolipids. In such combinations too, fructose can be very useful. Okay, so those are the functions of biological significances of fructose. Let's see the next one, that's about the significance of deoxyribose. Deoxyribose, as we said, deoxy is a oxygen where a ribose where oxygen is removed from the second carbon atom, okay, of 5 carbon atoms that are present in ribose. Now we are all familiar about the particular molecule called as deoxyribonucleic acid, or simply the DNA, which is a genetic material present in most of the organisms on this earth. Okay, and this deoxyribonucleic acid is formed of a pentose sugar, and the pentose sugar present there is deoxyribose. How it is different from ribose? We know in ribose is also a pentose sugar, okay, but the difference is that this ribose has oxygen in its second carbon atom, OH group is present, whereas in deoxyribose there is only H. Deoxyribose sugar is very important because it forms the genetic material of living organisms. Similarly, ribose is also important because it forms the structure of nucleotides. You have studied RNA, ribonucleic acid. So there ribose sugar is present, okay, in deoxyribonucleic acid. Deoxyribose sugar is present in RNA, ribose sugar is present, and this is present even in the viruses and bacteria, like you have seen RNA viruses. So their genetic material itself is a RNA. Or retroviruses, again they have RNA as their genetic material. Okay, not only that, these ribose sugars are also important components of nucleotides like NADH, NADPH and FADH2. So these about these you will be studying in glycolysis as well as in Krebs cycle. They will come a lot in those kind of metabolic reactions. Not only that, ribose is also part of ATP. Adenosine triphosphate, okay, adenosine triphosphate, in that this is a nucleotide, and again it is formed of ribose sugar, and combining with adenosine, okay. So adenine, that is the nitrogenous base that is present. When nitrogenous base adenine combines with the ribose, it is called adenosine. triphosphate, 3-phosphate groups are added to it. Now that's about the structure and its uses, biological uses within the cell or the body. In addition to that, deoxyribose is also used in the management of congestive heart failure. Okay, it can also be used in the treatments of cramps, pain, stiffness. Okay, usually that occurs after the exercises. Okay, so I'll just conclude the class for this now. Next we are going to see about oligosaccharides and polysaccharides. That will be discussed in our next coming class. Thank you.